Hi, I'm Jennifer Strande from the Medical College of Wisconsin in Milwaukee, Wisconsin. So I presented this morning on incorporating the person into personalized cell models. So we use induced pluripotent stem cells, or iPS cells, as a model system to understand cardiovascular disease. And a lot of groups are using iPS cells as model systems to understand genetic diseases. We're using them to understand other cardiovascular diseases that are caused from a multitude of mechanisms and maybe not have a genetic component. And so to do this, we emphasize the importance of clinical phenotyping of a disease population. And so we use a method called machine learning or unsupervised learning then to really tease out the population into clusters and identify the patients who have this disease who are most similar to other patients with that same disease. And we feel that starting with these clusters of patients then will improve the outcomes of getting a significant variable that is a contributor to that disease process. So once we identify the patients into clusters, we collect their blood samples and do proteomics to look at the differential changes of proteins in the blood of patients with heart failure with preserved ejection fraction versus the control patients. And from there, we have identified a protein called S100A8. And we take the recombinant form of that protein then, and we can put it on our iPS-derived cardiomyocytes then to study the functional effects of this protein. So once we have the protein on the iPS cardiomyocytes, we can do bioactivity screens like electrophysiological profiling or calcium transient profiling. And you can use any method of choice then to look at functionality in the iPS-derived cardiomyocytes. So when we did this in our assay, then we showed that the S100A8 protein then had adverse effects on the electrophysiological profiles of these iPS cardiomyocytes and also caused abnormal or delayed depolarization events. So it affects the kind of the beating tendencies of these cardiomyocytes. So we think that, because we did this extensive clinical phenotyping of the patients to begin with and identified a cohort that had heart failure with preserved ejection fraction who had a tendency to have atrial fibrillation, and identifying S100A8 as part of this disease process and confirming that in our cell culture model, then we may have identified a new protein then which is causal to developing heart failure with preserved ejection fraction in patients with atrial fibrillation.